virus, not just, it's important to know the knowledge about the virus. However, it's very important to do some acts about it. So, first of all, as my previous talk was about the characteristics of the coronavirus, however, I am focusing on the in this figure, focusing on the envelope of the virus, which contain three glycoproteins: spike, envelope, and membrane. These are proteins, actually, especially the spike one is very, very variable protein on the top of the envelope, which leads to combating the immunology and the immune system inside the host and leading to generation of several variants. So the replication cycle of the virus is very, very important. So first, if you can see, the virus can attach to its receptor on the surface of cell, mainly respiratory, sometimes digestive, sometimes renal cells, nephro cells. And then I'm focusing here to see about the genomic and subgenomic RNAs in this infected cell. So all the RNAs actually divided after the replicase. As it is positive sense single-strand RNA, it directly acts as messenger RNA inside the cell. So mainly the process of the coronavirus proteins here in endoplasmic reticulum and Golgi complex, as it is the assembly site of the virus and generating millions of the virus to go back. So this site actually is a site to combat the virus. I mean, for example, if you just inhibit some process here, so you can do something about assembly site and the number of the generated viruses. The genomic organization of the virus, as you can see here, it's exactly like normal coronaviruses, except for a very few ORFs here and non-structural proteins. They are exactly 16 non-structural proteins in the polyprotein, in replicase protein. It's starting from 5' untranslated region as a positive sense, and also a very big polyprotein as RNA-dependent RNA polymerase, and then the spike and the other non-structural protein, envelope, membrane, and nucleoprotein, and the very end is 3' untranslated region. So for molecular diagnosis, we normally target these genes actually, but it is some mechanism to target these kind of specific sites on the genome of the virus to target it in the molecular labs. So for this targeting, we should know about the phylogenetic tree of the virus. For example, here this is our normal coronavirus, which just yesterday they officially call it COVID by WHO, and we can see it is the most closest virus for it is came from a bat, so it's the nearest precursor of the virus. But I think this is going to be changed because still analysis is ongoing, and we have to wait more time actually to see the final analysis about the origin of the virus. So with the backup of those sciences, we can target the molecular diagnosis of the virus based on this workflow. For example, doing research and diagnosis approach, so by selecting any of the genes according to the multiple sequence alignment, and then targeting some of those genes to do primer design for both after reverse transcriptase PCR and a real-time reverse transcription PCR, and then doing in silico analysis to see these primers are exactly annealing to those sites, not to the other, for example, selected viruses. After that, starting for sample processing, which normally it starts with nucleic acid extraction, which is RNA, and then targeting the amplification of these RNA, extracted RNA, to say if it is positive and negative sample. And then we can do furthermore analysis by sequencing and identification of the virus. So the very important thing in this virus is actually the biosafety and risk management for the samples. As my colleagues talked about the outbreakable and the high number of patients currently in the world. So we have to be very precautious about this virus. I mean, for example, accurately preparing for the safety and processing of the sample inside the lab, and also doing the molecular diagnosis in very precise way to not to be another way for infection actually. So, if you can ask one question, can we establish this approach here in Slemani? I think yes, we can, and we have to do it actually as an emergency lab. And thankfully, the General Directorate of Health in Slemani, in collaboration with Directorate of Veterinary, have already started this approach to establish an emergency lab in Slemani, and the works are ongoing. Hopefully, going to be declared very soon. So, as we said, we can plan for the lab as an emergency lab, as a design here, to detect and diagnose the novel coronavirus in our city, and also even verifying the protocol and also towards the identification of the virus by further sequencing and data analysis. The technical workflow of this procedure can be started as sample processing and RNA extraction, as I told, and then cDNA synthesis for normal PCR and also RT-PCR and real-time RT-PCR for normal detection of positive and negative samples. So targeting the amplification and result analysis after this amplification, and then declaring that if the case is positive and negative. And as my previous talk talked about this, it's very important actually, because this molecular approach can diagnose, can detect the virus even if it is in its incubation period and in the very first day of infection. After that, the confirmation and identification can be done by sequencing analysis, and we have a variety of methods to do, either by standard sequencing or next generation sequencing, and doing result interpretation, and even submitting our results in GeneBank, which is very important for our country. To summarize my talk and what we have said about this topic is our understanding to the genome of the virus, and also the proteins from the overview, and also the replication cycle, where it's exactly ongoing inside the cell, and what we can target, what genes we can target for the virus, and then how to design molecular diagnosis of novel coronavirus, and biosafety and risk management, and also the technical procedure of the procedure. Thank you very much for your attention. Thank you. Sorry, this is my acknowledgement to the sponsor of this talk. Thank you.